Okay.
So, uh,
No more present.
Burgos.
Okay, thank you. Thank you so much, Ricky.
Hello. So thank you,
Uh,
ma'am, Burgos.
Thank you so much, Ricky.
And then we'll proceed with the next presentation. Again, from UPB. And then it's going to present to us by Alex Regalado, which is she's going to talk about the Fortunus pelagicus. Go ahead, ma'am.
Hi, Sir Ting. Can you hear me? Everyone?
Yes, I can hear you.
Okay. So I will share my screen.
Yes, please.
Does it show already?
Yes, ma'am.
So, okay na. So to start with, I'm Alexandra Bagarinao-Regalado from Ocean Biolab, UPV Science, and today I will present our work together with Dr. Wilfredo Campos on the distribution patterns of early stage crabs in Northeast Panay, Central Philippines, providing insights on the significance of seagrass and mangrove habitats on the early life stages of Fortunid crabs. So I will start with the established fact that many coastal species utilize seagrass and mangrove areas during their early life stages. So it is believed that the physical structure formed by the root systems of mangrove trees and the density of blades in seagrass beds offer not only refuge from predation but also abundant substrate for diverse assemblages of epiphytes, which in turn serve as rich food source. So Brocurian crabs generally prefer structurally complex habitats as settlement sites. So for example, blue crabs mate in shallow coastal waters, for example in seagrass beds, then migrates offshore to spawn. So the zoea stages can be drifted up to 80 kilometers and can be found in mid to offshore areas. Then the Magalopa inhabits the sheltered uh, coastal waters in seagrass beds and look for settlement sites where it will grow. So these structurally complex habitats are found along the coast of northeastern Panay, where sheltered embayments, mangrove forest, seagrass beds, and coral reefs are found. So this may explain the thriving blue crab fishery in the area. So, however, blue swimming crab fishery in the Western Visayas Sea shows a decreasing trend in catch per unit of effort and experiencing overfishing in the last several years. So, moreover, northeastern Panay is currently facing serious deterioration of coastal habitats, such as intensified agricultural activities, coastal development, excessive sediment concentration, resource consumption, including unmanaged fisheries, which leads to significant habitat alterations affecting the settlement and recruitment processes of early stage crabs. Since these areas are important habitats with strong influence on recruitment and dynamics of crab populations in the Visayan Sea, the spatial distribution and abundance of early stage crabs along the northeastern Panay coast was assessed. So for the study area, survey sites cover representative areas of the northeastern Panay. These are Bangkal Bay, Concepcion Bay, and Banati Bay. So there are six light trap stations per bay, and 5 plankton tow stations per bay. So night sampling was done from November 2016 to April 2017, covering the peak spawning period of Portunus pelagicus in the Visayan Sea. So zooplankton were sampled by means of 5-minute surface horizontal tows using a rectangular frame net and a flow meter was mounted at the mouth of the net to measure the volume of water filtered. So this figure shows the light trap we used in the experiment. And we modeled our light trap design after that of Rogner et al. 2003. And light trap were set in two different habitat types. One with seagrass bed near mangroves, and one is seagrass bed only. So 3 replicate traps were deployed per habitat type, and traps were deployed during the incoming of the tide in no more than 3 meters of water depth for a period of 3 hours. So light trap and plankton surveys were done at night during new moon to take advantage of spring tide and maximize the effect of light trap. So for light trap site selection, we focus on those areas because previous studies have shown that early juvenile Pertunus pelagicus densities are highest in seagrass beds nearest to mangroves. So all samples were fixed immediately in 10% buffered formalin solution, and Brachyuran, Zoea, and Megalopa were identified and sorted out, and density were computed. So this figure shows the main morphological characteristics used for the identification of Brachyuran, Zoea, and Megalopa. So for the results, this figure shows the top 10 zooplankton major groups caught by horizontal toes, shown in the lower left, and light traps deployed in seagrass beds near mangroves, indicated by positive MNG, and light traps deployed in seagrass beds only, so indicated by negative MNG. So results for Concepcion and Bancal Bay is also shown here. So the calanoid dominated the plankton community in Concepcion and Bancal Bay, as highlighted by the red box, whereas shrimp dominated Banate Bay. So crab, zoea, and megalopa are abundant, included in the top 10, both in Bancal Bay and Concepcion Bay. They are also present in Banate Bay, but are not in large numbers. So this figure shows the spatiotemporal distribution of crab zoea densities in Northeast Panay. So abundance was highest during the onset of Northeast monsoon in November and December. So for spatial differences, sorry. This coincides with the peak spawning season of Pertunus pelagicus, which occurs from October to April. So for spatial difference in abundance, Bancal Bay recorded the highest, followed by Concepcion Bay and Banate Bay. So this figure show that spatial temporal distribution of crab megalopa densities in Northeast Panay. So same pattern was observed with the abundance being highest during the onset of Northeast monsoon in November and December. And for spatial difference in abundance, Bancal Bay and Concepcion Bay are comparable, while Banate Bay had the least number of megalopa. So this figure shows the overall mean density of zoea and megalopa across 3 bays. So the difference in abundance across bays is significant, with Bancal Bay having the highest abundance of early stage crabs, followed by Concepcion and Banate. So the abiotic parameters collected during plankton and light trap sampling are comparable and may not clearly explain the differences observed. However, Bancal and Concepcion Bay may be more structurally complex in terms of high seagrass cover in the area as shown in the table. So Bancal Bay Carles has a very extensive and continuous seagrass bed. Concepcion has several grass beds but are not continuous, while Banate Bay has a very patchy grass bed with low seagrass cover. So the coastal resource map of the province of Iloilo shows the same, with the highest seagrass cover in Bancal Bay. So the area shaded by cyan blue color indicates seagrass beds. Mangroves are shaded violet, and blue fish ponds are shaded light blue. So as mentioned earlier, that Banate Bay consistently recorded the lowest abundance for both zoea and megalopa. This may be associated with its characteristic patchy seagrass bed, and significant alteration occurring in the area where a large portion of its mangrove forest have been converted to fish ponds, which in turn may have contributed to excessive suspended sediment concentration in the area, as indicated by turbidity measurements recorded by campus. So next. This figure shows the result of our experiment on differences in abundance of early stage crabs in two different habitat types. One is seagrass bed only, and the other one is seagrass bed with mangroves nearby. So for horizontal toes, the density of Zoea and Megalopa are higher in seagrass beds near mangrove in Concepcion and Bancal Bay. For light trap, density of early stage crabs are higher in seagrass beds near mangrove only in Concepcion Bay. So this conforms with the result of previous study on independent trawl survey in seagrass beds of Concepcion, Iloilo, and Talibon, Bohol, where highest abundances of recruits in seagrass beds nearest to mangroves. So in summary, although we have not identified the early stage crabs collected into lower level or family level, the independent trawl survey of Campos shows that Pertunid crabs, including the most exploited Pertunus pelagicus, dominate the area. So our study suggests that these seagrass beds are critical habitats with strong influence on recruitment and dynamics of crab populations on the Visayan Sea. And these habitats, Bancal Bay and Concepcion Bay, are critical to the settlement of crab host larvae. So this may help improve the management and sustainability of crab fishery, particularly the heavy fish stocks of blue crab Pertunus pelagicus in the area. So I would like to acknowledge Ocean Biolab, University of Philippines Visayas, and our funder, the OVPAA, and Dexter Pilota and Kim Nunez for field assistance. Thank you very much.
Uh,